Welcome everybody for joining us. This afternoon we have Dr. Will Greenland from Agilent. He's going to be talking about protein glycosylation. So just to give Will an introduction, Will's a chemist. He began his research career as a development scientist working for Northeast Regional Thames Regional Assay Unit, Immunoassay Unit. And in 1992, he joined R&D Systems, where he worked in a number of different roles, and he's worked on enzyme conjugation, novel biologicals, peptide labeling, and proteomics. While still at R&D Systems, he did his PhD in 2002 at Canterbury University, where he worked on imaging technologies in cancer and bone disease. And then in 2003, he joined Agilent Technologies, where he's been ever since, and he's been working on mass spectrometry applications and mass spectrometry hardware. And I'll hand it over to Will, and it's a pleasure to have you talk to us today.
Thank you, Rod. It's a real pleasure to be invited to give this talk. So I'm always very happy to talk about mass spec for sure. The talk today is focusing on glycoprotein analysis by LC-MS, and this is we're going to look at several things to do with that. So I am Will Greenland from Agilent, and one of the things I do specialize in is also biopharmaceuticals. I don't have a PowerPoint presentation because I don't like the linear format. So I do prefer more interactive approach, and I encourage questions at any time. Can everyone see me?
Yes, I think they can.
Okay, good. So as I invite questions at any time. So glycans, we're going to just share a my screen with you with any luck. Okay. Right, share screen. Okay. So this is where most people start with any sort of scientific work. These days, the Wikipedia page, and here we have the entry for N-link glycosylation. Glycans are important in proteins for a number of reasons, including protein folding, fine-tuning of biodistribution. They are found in pretty much all organisms, including plants, insects, and mammals. As we can see on the right-hand side there, they tend to get more complex in higher species like mammals versus simpler things like plants and yeast and insects perhaps. But they can be very complex for sure. If we look at humans, some proteins have no glycosylation at all, like things like human serum albumin. Some proteins have some glycosylation, like monoclonal antibodies have a couple of glycans, mainly in the hinge region. And then we get some, again, serum proteins like fetrin, which has lots and lots of glycosylation. So we find them in varying amounts in different proteins. Biology doesn't put these things in for fun. They are generally doing something. So if you are interested in protein interactions, metabolic diseases, biopharma, you probably need to have a look at glycosylation at some point. This makes it a very big subject, which is good in some ways, because it means you can spend your whole career doing it. It does mean there's a lot to get your head around for sure. So to kind of quote Rod a bit, it is a difficult subject and often ignored due to its complexity. So we are going to look at, particularly today, I'm going to be focusing on what is referred to as N-linked glycosylations. These are a post-translational modification of course, and they are resident on asparagine, the amino acid, in a motif which goes N, X, S, or T. So either on a there's a certain sequence of amino acids which is required for an O-link glycan to form. I'm also going to be mainly focusing on recombinant proteins, so proteins which are actually grown in cell culture and purified. I think we got someone who needs to mute
Could
themselves.
I ask all the participants, if you're joining us, can you please hit the mute button, please?
Sounds as if someone is underwater. Okay, so we're going to mainly talk about recombinant proteins. If you go and look at the Wikipedia page there, you can kind of see the basic structure. Towards the bottom, you've got two blue squares, and these are the glucanacs, which are directly linked to the asparagine, and then the green circles are a branch structure of mannose or hexose, depending how sure you are which you forgot. Okay, fine. I'm just going to bring up a second slide whilst I carry on talking, hopefully. So, share screen and share. Okay. So, there's several ways to look at glycans on the proteins, depending on exactly what you're trying to find out, and also the complexity of the system you were looking at. The first way is on the intact protein. And I'm going to mainly be using a few graphics from the paper that Rod and I and Tiago and others at Oxford recently published, based on the COVID-19 spike protein. And this is one of the graphics we've used. This is an intact protein. It's actually the receptor binding domain, which is a small part of the complete or intact spike protein. It's probably about 1/6 the size of the total protein, and it only contains 2 glycosylation sites. So this is really what I would describe as simple glycosylation, and it's already becoming quite complex. But we can still figure out what most of these peaks are, with a little bit of common sense and knowledge about how glycans form, and the most likely species of glycans we are going to get. So intact analysis is a very good way of getting an overview of your total glycosylation for relatively small and relatively simple proteins. The difficulties occur when this becomes more complex. So the full length spike protein actually has 22 glycosylation sites versus the 2 here, and each one of those sites may have 1 or 2 or 5 or 6 or more different glycans. So the trouble with that is there are now millions of different combinations of glycans on a single protein. As soon as we try and analyze that on an intact level like this, we basically get so many peaks. They all kind of merge and smear and actually disappear into the background. So as soon as it gets very complex, it's no longer possible, at least with current technology, to do this sort of analysis in the intact protein. So then we get two choices. We can either cleave all of these proteins off, glycans off, and this is what's known as a released glycan analysis. For the end-linked glycans, we use a glycosylase, which is called PNGAs, and this cleaves off the glycans very nicely, and we can analyze those in a VMAS spec. The trouble with that, though, whilst it gives again an overview of the total glycosylation, we also lose the site-specific nature of the glycan. So we don't know which site any particular glycan was on the original 22 sites on the full-length spike protein. So whilst it's useful, you're kind of losing a lot of potentially valuable information. So my preferred option at this point is actually to look at the protein on a glycopeptide level. And this involves doing some sort of enzyme digestion, often with trypsin, and then that gives us an opportunity to look at glycans on the specific peptides, so we can tell what glycans are on which sites on the original protein. However, this is not a trivial matter by any means. We're just going to have a quick look at some of those. I'm going to stop showing this one, hopefully pull up the sequence of okay, so you should be able to see the sequence of full-length spike protein, and I've highlighted the end-linked glycosylation sites in red. You can see here that there is a particular motif. It always starts with an N. It always finishes with an S or a T, and then we can have any amino acid in the middle apart from P for proline. If we did a triptych digestion of this full-length spike protein, we get something which looks like, I'm going to have to, this is it. So we get something which looks like this. So we get one bunch of triptych peptides. That works pretty well up to a point, but we then, if down the list, we are seeing some peptides which actually have more than one glycosylation site within the triptych peptide, and towards the bottom there we've actually got a peptide which has 3 different glycosylation sites, and is also very long. these get increasingly difficult to identify in the VLC-MS. It may be an appropriate technique for a completely unknown protein, but where we know what the protein is, I actually believe there's a better choice here. And what we did in the paper we're publishing is we used elastase. I'm going to scroll down. I tried a few other enzymes as well as you can see. And an elastase is not a typical protease used in proteomics, mainly because the peptides are too short for classic proteomic identification, and also it's relatively non-specific, which makes proteomic identification almost impossible due to the high false discovery rate. However, when we know what the protein is, those disadvantages disappear, and generally it's easier to identify something small than identify something large. We also found that we didn't appear to get any peptides with more than one glycosylation site, so it seemed like a good choice for the work we were trying to do. Okay, just going to go, hopefully stop showing that and try and share something else. Going to go back to the paper. I'm going to scroll up just to give you an idea of some of what was required. So this is again just a graphic from the paper, but what it's showing us is some of the peptides we found in the elastase digestion, and it also shows you some of the glycans we observed and also inferred as well. I shall get back to that inferred part a little bit later. But it's also to scroll down again. I wanted to show you this is this graphic is it's a chromatogram, and it's actually showing us just one of the 22 glycosylation sites on the intact spike protein, and it's just showing you just how complex and the number of glycans we've got. I can't count them all up, but I think there's probably at least 20 there, so 20 different glycans just on one peptide. It does make it complicated for sure. Okay, so if there's been obviously an explosion of interest in the glycosylation spike protein in the last year, and there's been some very good and interesting publications come out of that. They've all kind of used the same basic technique, which is to use a very expensive Orbitrap from Thermo. They are all using some of it between 3 and 5 different enzymes to cleave the protein into its glycopeptides, and they are all using nano LC. So this is a very low flow rate chromographic techniques, something like 300 nanoliters per minute, and they are all using nano ESI and some sort of auto SMS to identify glycopeptides, and then some fancy software to analyze the data. And that all works very well. But like all scientific techniques, there are some problems with this approach. Each run is approximately 4 hours long. It's a long time to be collecting data. Typically, the percent RSD on a nano ESI is about 20%. So it's really hard to get reproducible data. You only see the things which have triggered an auto MSMS cycle. So anything which is present but hasn't triggered auto MSMS won't be seen, which is a problem. But the main thing is I would describe this as a proteomic approach, and it's very good for the discovery of interesting glycopeptides for sure. What it's not good for is to compare 2 batches of recombinant protein in any sort of quality control, and it's not so good for any quantitative or semi-quantitative analysis of relative glycans. It's interesting how some of the data has been reported in about they've avoided any absolute quantitation at all, and everything's been reported with a relative amount. And I do think some of the data has been over-interpreted, perhaps, and I also think there's maybe an over-reliance on some of the software which has been used to identify some of the glycans. But there is no such thing as a perfect technique for sure. When we looked at this, we decided to take a very different approach to solving the problem. So rather than a classical proteomic approach, we went for what I would describe as a biopharmaceutical quality control approach. So what we were looking for was a much more reproducible method. So one of the first things we did was to switch to an analytical flow rate HPLC. So we are using, I think, about 0.3 mils per minute instead of 300 nanoliters. So about 1,000 times more higher flow rate. There is a mindset that you can only do this proteomic or peptide approach with nano LC, and that's simply not true. The only reason to do nano LC is if you are sample limited. So if I've pulled out a few brain cells from a biopsy, or I've got a dissection or something, or a tumor or something like that, where I have a very small amount of sample, I'm forced to use nano LC in order to get enough sensitivity out of the system. I'm essentially trying to keep the sample concentrated by using very low flow rates and internal volumes. However, if I'm not sample limited, and this is generally true with biopharmaceuticals, because we can grow as much as we want to, you're much better off using an analytical scale, HPLC. And the major reason for this is simply just more robust and more reproducible. Let me try and I'm just going to change the graphic if I can find what I'm looking for. So I'm going to. Okay, so this is actually some of our chromatography. So we got some really nice, beautiful peaks there. These are all glycopeptides from the full-length spike protein. What we can see here is we also decided to use reverse phase, whereas when they're using when they're doing just re-released glycan, people would typically use a helix column or something else rather than reverse phase. Reverse phase is very good for separating things based on their hydrophobicity, so it separates the peptides really well. What it doesn't do is it doesn't separate the glycans. So because the glycans are very hydrophilic, they tend to have the same reattention time. But what we did here was we used this, rather than perceive this as a disadvantage, we decided to use it as an advantage. So if we look between about 46 and 47 minutes there, and also 48 and 49, it's the two larger kind of purple peaks to the right hand side. If you look closely at those, there's a number of peaks on top of each other. And what this is actually different glycans on the same peptide. So the retention time is basically controlled by the hydrophobicity of the peptide, and then all of the different glycans on that single peptide tend to have more or less the same reattention time. Okay, so what we figured out fairly quickly is that once we've identified a peptide and a glycan at a specific reattention time, so we got retention time and a mass. We don't need to keep on and doing auto SMS. We don't need to keep identifying it by auto SMS on each run. We know where the retention time is going to be, and we know what the mass is going to be. So as long as we are armed with that information, we can actually construct a database, and we don't need to collect any data beyond simple MS1. Okay, and that is what we did. So that is kind of what we've done. That's kind of what we've created here, and this is essentially a printout of the database. Now, this then led on to, so what we then realized was we can create the database from glycopeptide we have observed and identified, but we can then also add glycopeptides, so different glycans on peptides, which we haven't actually seen yet, because we know what the retention time is going to be roughly, and we know what the mass is going to be. So if we look, let's try and find a good example. A few too many. If you look to the top left-hand side, we've got the glycans which on our peptide NL/IT, and to the bottom of those we've observed man 8, man 7, man 6, man 5, man 4, and then there's also some man 9 at the top. We can then we've inferred that man 3, man 2, and man 1 would be at a very similar retention time, and we can calculate the mass for that. So we can basically construct a database, not only on the glycopeptides we've seen, but also what glycopeptides might be present in other batches of recombinant protein. So this is it's quite a powerful way again for comparing different batches of recombinant protein and seeing whether changes in the cell culture make a difference to the glyco expression. Okay. And in order to help us with that, we have to find it. We know that scrolling, further scrolling. So you do need to look up how glycans are processed in biology. So we have in the center there we have the processing order. They all start off as high mannose glycan, and this gets processed to a what's known as G0, and G1, then G2, A1, A2, and then B beyond that. So by understanding the processing order, we were able to have a good. I'm trying to avoid using the word guess. We were able to infer, for instance, if we had a man 5, we would also automatically look for a G0 and the G0F. If we found one of those, we would look for a G1F. If we found one of those, we move on to the next stage, and then the next stage, and then we added those to the database as well. So it becomes quite a powerful tool. Appreciating both the chromatography and the fact that glycans are predictable to some extent. Okay. So that's kind of how our database works. Just to fill that out a little bit more, we did have to identify the glycopeptides, and that's actually quite difficult. So it's difficult because you have a mass, and that mass contains an unknown peptide and an unknown glycan. If we try and match that mass to a theoretical something, basically I tried that, and the false discovery rate was about 95%, so ridiculously high. What I found was that as long as you can identify either the glycan or the peptide, that gave us the mass of the other half, and then the false discovery rate became manageable, and we can do something with the data. This turned out to be quite a manual process, and on the screen now is a good example of how we did that. This is a beautiful spectra. It's a spectra of the a relatively short peptide from the receptor binding domain, and what we did was a soft fragmentation, and this is given as a beautiful glycan ladder, very clearly demonstrating this is a man5 glycan attached to this peptide. Because I could identify it as a man5, I could work out what the mass of the peptide was, and then I could compare that to the sequence. That still leaves a bit of room for false discovery. So what I then did with not all of the peptides, but about a handful now, is I also did what's free experiment. This is a bit, it's not a usual technique. It's a way of creating fragments of fragments in a system which isn't really designed to do it. So it's, this was done in an adjuvant QTOF. The information has basically, if you know what to look for, and this is essentially a manual. It's not. I wish I wouldn't quite describe it as de novo sequencing, because we kind of know what the sequence should be. We are just confirming that by looking at the MS3 data here, and that actually gave a really nice match for this peptide. So now my false discovery rate on this particular glycopeptide is pretty well zero. Very confident in its identification, which is always nice. And then just at the bottom there's a table of the ions and their assignments, and also the ppm errors here, which are all nice and low, which again gives me great confidence in the identification. So we kind of went through this approach for the rest of the, well, as many as we could, and we produced the database, which again is up the top here, and we managed to cover about 2/3 of the glycopeptide sites with the elastase digestion. I am currently working on looking at the tryptic digestion, also the chromatographic digestion as well. And I was doing some of that this morning. So I'm going to try and share that screen. So I'm going to stop that. And so this is some of the tryptic data. Here, if you look at the bottom, we very clearly have the beautiful this is actually a man8 glycopeptide. That's really beautiful data. We can also kind of see again some of the difficulties in analysing this sort of data with the middle spectra there. I'm going to zoom in on the bottom bit to begin with. So it's the thing which says +4 kind of in the centre of the screen there. So this is a glycopeptide. This has a +4 charge state. To the left-hand side, we have the protonated adduct, but we also have got a potassium adduct and also a sodium adduct, and actually a mixture of both. So we're not just talking about one single pretty peak. And if I just zoom out again, if we look at the main peak there, this is the triply charged version. Again, this is just one glycopeptide, and we have got a bunch of peaks here. And if we zoom in, I'm going to zoom in on just the protonated adduct for hopefully simplicity. Even with the protonated adduct, we have multiple peaks. The very first one there on the left-hand side is what I would refer to as the monoisotopic peak at 1175, and then the other peaks are basically due to the carbon-13 abundance in the isotopic distribution of this data. So even for one glycopeptide, you were dealing with multiple peaks, multiple adducts, and multiple charge states. And this makes the data analysis very challenging. And each one of the coloured peaks towards the top of the screen there in the chromatography has similar lower data. So it does require some software to analyze this for sure. I always like to double check the software is doing what I think it's doing. So I do think it's important to apply a manual analysis to all data at some point to double check it. And I actually found it was very important for the data analysis part to actually be able to do a lot of the maths myself. Okay, I can see I'm at 45 minutes. I shall be wrapping up shortly, so don't worry, Rod. I'm just going to perhaps do one last thing to show. Not even sure if Rod's seen this yet. So this is it's obviously an Excel spreadsheet. I might just make it a bit big if I can. Hopefully you can see it. So this is an Excel spreadsheet, and I was using quite a lot of these to analyze my data. There's a whole list of compounds down the side here with the masses and the tension types. So this is some of the raw data which I pulled out. If we scroll down on that list, if memory serves me right, there's about 6,000 lines. So to manually try and interpret that data is a little bit tiresome to say the least. But what you can do if you like this sort of thing is you can write about a macro to do it. So I just click run now. And what this is doing is basically looking at the masses and the retention times, and it's trying to pull out possible glycan adducts. So it's just trying to identify things which have a mass separation which corresponds to a glycan, and lo and behold, it does it very, very nicely. So we've got things which are possibly a difference between a hex, which is generally a mannus, and a bunch of other stuff. The first thing I did with this though was to run it against data which had no glycans, because I was interested in what sort of false discovery which I got. So I ran against BSA and lo and behold, it does pull out things which look like glycans. What it didn't do is it didn't pull out more than 2 masses. So if I was going to use this data, what I'm looking for is basically 3 rungs on the glycan ladder, and then I found that was enough to make the false discovery rate low enough to warrant adding it to a database. So again, one of the points I'm trying to make is there is I think there is an over reliance on black box software to do the analysis for you, and this can lead you down the wrong path. I'm very interested in false discovery rates all of the time, and I've tried very hard to make sure that the data we've published is as error-free as possible. But it is complex, and it is certainly challenging. Okay, I'm going to click on stop sharing there. So I think you can. I'm assuming that you can see me.
Yes,
excellent. I shall invite questions at this point.
Okay, so if anyone has a question for Will, please unmute and feel free to ask. If you would like to type your question in the chat box, I can happy to ask that question for you. While we're waiting, Will, I mean, yes. I noticed from the spreadsheet that you were showing us, there's something like 4,000 compounds in the LCMS run. Would you like to comment about how you filter that data, looking for potential glycopeptides?
The first filtering step was really just to reduce noise. So I take out the very small peaks, but also as we saw from the multiply charged stuff, a peptide or a glycopeptide is invariably multiply charged. So any compound which just exists in a single charge state is likely to be a small molecule contamination, and can be removed pretty well straight away. So the charge range I think I was screening for was anything between 2 and 5 charge states, which is typical.
Did you obviously there are a large number of glycans present on Spike? Did you find any glycans that you would describe as unusual?
We did find a what's known as a bridging glucnac. I'm just going to see if I can share that. I don't think I actually draw. We might have the picture, we might not. So if we look on the right-hand side there, if below G0 we've got something which we've assigned as G0 plus glucnac. The trouble is we don't actually know exactly where the extra glucnac is, whether it's on one of the branches or attached to the first green circle, which is the mannus. we were finding these in quite high abundance, associated with MAN5 and also G0F, which makes us suspicious that it could be what's known as bridging glucnac. These are interesting because they have been implicated in immune evasion in some of the studies. So that could be contributing to the glycan shield of spike protein.
Just to comment, I showed that structure to David Harvey, and his comment was that we would never get that glucnac added onto the end of that chain. That would not
Excellent.
be normal. Yeah.
Okay. That's probably true. One of the things we need to be careful of is you can get some in-source fragmentation in a VMS. So as long as it has a slightly different reattention time, then I think that is a valid interpretation of the data. And I do remember we checked that. In fact, I think we've actually got it in the here. Yes, so we've actually got the glucnac there, the extra one on a G0F, and it obviously has a different reattention time to most, well, the other glycans. So that's a good indication. It is a bridge glycan. It's quite difficult to construct an experiment even with MSMS to determine that for sure. It might involve some other enzymes to check it. I didn't really think much beyond just observing it as something interesting.
Sticking on the subject of enzymes, elastase, this is an unusual choice. Yes. For your protease. Would you like to comment on the elastase, on any advantages you have with elastase?
It is an unusual choice for a proteomic approach. I think it's a very appropriate choice for biopharmaceutical approach. We did observe something interesting in that a lot of the glycopeptides seem to end on the C terminus, and that's at the right hand side. A lot of the peptides would finish with exactly the typical glycan motif. So they end in NXTUS. We are curious as to whether this is something about elastase and glycopeptides, whether for this is something which it tends to do. It might just be to do with how I started searching the data. As soon as I started finding several of these, I started looking for them harder. So as soon as you start looking for something, you tend to find it. Looking at both the triptych and the chiro data, I am seeing a lot of very unusual cleavages and what might be described as non-specific cleavages. So many in 3 different enzymes, but I'm starting to think that there's something about highly glycosylated proteins which make these proteolytic enzymes cleave in unusual ways. This is something which I I would very much like to have a closer look at, but I do suspect it's someone's PhD project that it's very hard to find the time with everything else.
That's a very interesting note to end on, Will. I think if there are no more questions, it just remains for me to thank Will for his presentation, to thank you for listening, and just to say, if you'd like to join us next week, we'll be talking about quantitative mass spectrometry. And if you are willing to unmute and just put your hand together and tell Will your gratification for his presentation. Thank you very much.
Thank you.
Thank you.
Pleasure.
Thanks.
Cheers. Okay.